So what I would like to do is I would like to invite our speaker, Dr. Armin Szegedi, who's been very gracious to come all the way from New Jersey, including having to wait 5 hours in the airport there. By way of an introduction, Dr. Szegedi, who is an MD PhD, who is currently the section head of psychiatry within the Division of Clinical Neurosciences and Ophthalmology at Merck and Company. And in this regard, he's responsible for the scientific design and oversight of all global clinical development programs in psychiatric indications within Merck Research Laboratories. He heads an international scientific team operating from research facilities located in the United States and in the Netherlands. Now, prior to this position, from 2008 to 2009, Dr. Szegedi was the therapy area head for the central nervous system area of global clinical research at the Schering-Plough Research Institute, and there he received the highest honor of the Chairman's Award from the CEO of Schering-Plough. He assumed this position, as I said, in 2008 after Schering-Plough acquired his former employer, Organon Biosciences Company, which he joined in 2005. So as you know, what happened was Organon essentially merged with Schering-Plough, which ultimately merged with Merck. So if any of you question Dr. Szegedi's loyalty and commitment to an organization, he has been at the same organization really for all these years. Now, while he was at Organon, he led a team that worked on several Phase 2 and Phase 3 projects in various CNS indications, including what I'm most familiar with, and that's a successful NDA submission for the novel antipsychotic drug asenapine for the indications of schizophrenia and bipolar disorder. He also led a global clinical development team on a broad spectrum of other neuroscience indications such as depression, alcohol dependence, ADHD, and cognitive disorders. Prior to 2005, Dr. Szegedi was in academia, which is near and dear to our own hearts. He received his medical degree from Johannes Gutenberg University of Mainz in Germany. And had a very high-profile, successful academic career in clinical university research settings, including a full professorship in clinical psychiatry and deputy chair at the Department of Psychiatry and Psychotherapy at the Medical University of Berlin. He's a psychiatrist and psychotherapist by training, has extensive clinical experience in the treatment of patients with a whole spectrum of adult psychiatric diagnoses, and over more than a decade, he's conducted clinical research in academic settings in various psychiatric indications, including designing, conducting, analyzing, and reporting clinical trials in a wide spectrum of neuroscience indications. He has published many, many peer-reviewed publications and book chapters, and has really been a thought leader in the field of psychopharmacology for many years. So it's my great pleasure to introduce Dr. Szegedi, and I very much look forward to his talk. Thank you.
Well, thank you. Thank you very much, David. This was probably one of the most beautiful introductions I've ever had. Thank you very much for that. I would like to add one point, which is maybe also important to understand a little bit where I'm coming from. I studied theology before I studied medicine, and the common denominator was that I was always interested in human beings and what makes them think, what makes them act in the way they do, and particularly also I was always worried about suffering. I had a chat this morning with John about this common denominator, and that was finally something that drove me into psychiatry, to become a psychiatrist. But while all these things had a very psychological or spiritual component, one thing that I didn't expect on my way was that drugs can make a huge difference in this area. So that was the defining moment for me when I was an intern doing my MD thesis at the psychiatric department, interviewing 150 patients with a structured interview. That I saw, I had to interview them at the beginning, at the end, and I saw incredible changes in these people with schizophrenia, bipolar disorder, and I realized that actually the drugs were strongly responsible for that. So at that time point, and since ever then, I became very passionate about developing new drug treatments, and that is the reason why I finally also went into industry to do what I'm doing right now. So what I'm going to share with you. So first of all, my disclosure has been done beautifully by David already. Sorry. Okay, should I stay rather here? Maybe it's okay. Yeah, I can do it. No problem. Let me take that from you. Okay, sorry. Like this? Okay, good. So I just want to point out that I'm here as a clinical scientist and not so much as a representative of my company. So the views that I'm sharing with you here today are more my thoughts, which have emerged actually in the last few weeks. And there will be also a lot of remarks which may be not so enthusiastic but rather worried about the future of our area. And I'm going to tell you a little bit why I'm worried. So in the first part of my talk, I'm going to talk about the process of drug development as it happens right now in industry, and also partially with involvement of academic institutes, and also to have a critical look at the productivity of R&D development in the last few years, and then turn in the second part on specific challenges which have, in a way, plagued particularly psychiatry as a field for drug development, and particular challenges which I see going forward, which may be perils for progress, as I called my talk initially. So let me start with something that is truly quite new, and it has to do with headlines which relate to big pharma and measures that have been taken just about in the last 6 weeks. So Pfizer plans to cut billions in annual R&D spending. That was February. Glaxo to stop pain and depression research. February, 2 days after Pfizer. Sanofi Aventis chops into R&D budget. Just 5 days later. And recently, just a few days ago, AstraZeneca to drop psychiatric research. And when you look at the last one, that was the one that particularly worried me. The decision to drop psychiatry drug research reflects the unpredictable and risky nature of clinical trials to assess medicines working on the brain, as well as a lack of good scientific opportunities. Of course, these press releases always have some kind of a coded language, but I think there is a lot buried in here which I would try to touch upon to make it more transparent, what I see as areas where we have to work together in the future if we care about this area of drug development, which I personally do. So the path of drug development, as you know, is a very lengthy one, which includes a lot of components. In principle, there is a lot of efforts happening in the phase which we call in the industry the preclinical and discovery efforts, going from synthesizing novel compounds, looking at their pharmacological properties, looking at toxicology, screening for targets, looking at selectivity, selecting the right compound, and then looking at PK/PD of these drugs, and finally looking into animal models where the drug acts in a way that hopefully is predictive of a success in humans. Then you have usually this big step into first-in-human, phase 1. In many companies right now, there is also another component attached to that, which our former R&D chief called POC Light, which basically is in a way proof of concept, but not really proof of concept. You have almost proof of concept, which gives you a little bit of more confidence that finally you can go forward, spend more money, get proof of concept, and then do dose range finding, and then going into pivotal trials and long-term safety trials, and then hopefully you file your NDA. As you can see, the whole thing takes you at least a decade, usually a decade and a half, and it's a very lengthy process in which a lot of effort is involved. I actually was very much surprised when I learned from the inside in industry how many people actually work on a single compound like asenapine. When I first read the protocols and the program of asenapine 5 years ago, I thought, oh, this is a no-brainer. This is going to be approved easily. In fact, I spent probably 70% of my time in the last 4 years in getting this drug approved at the FDA. So it's really a huge, huge effort. When you look at what I call the Via Dolorosa of drug development, you start on average with 5,000 to 10,000 compounds in drug discovery, and over the years, you hopefully end up with one that finally gets approval. At the same time, the number of humans involved in basically proving safety and efficacy of the drug exponentially grows, so that in the meantime, usually you have to submit dossiers with about 3,000 patients per drug in order to have a reasonable safety database. Where do you spend the money? Roughly, you spend 1/3 in preclinical, which is already a lot of money. You spend about 1/2 of the overall costs up to phase 2, and then you spend additional 1/3 in phase 3, which is usually very intensive, labor intensive, and involves a lot of costs for our specialty for clinical research. That is the big chunk of costs that are being spent. Now, how have the budgets developed for R&D in the last year? So you can see here the blue bars. Those are the investments per year, the total R&D investments of pharmaceutical companies as organized in the Pharma consortium. And in the last few years, also numbers have become available for all pharmaceutical companies worldwide. And what you can see here is really a exponential growth over time. So in 2008, it was around $50 billion annual budget spent in R&D by the pharma companies. When you put into context the NIH budget in the same time, so here you can see in orange the bars that I showed before, and these are all biopharmaceutical companies worldwide. You see that the NIH budget is also substantial, but it plateaued off over the time in the last years. And when you break NIH budgets down, so here now the blue bars are the total NIH budgets, and you look actually at clinical research, which is the light blue, and you look at NIH clinical trials, then you see the bars become smaller and smaller and smaller. And finally, when you look at NIH neuroscience, the budgets for neuroscience overall are about $5 billion per year, but the majority is not being spent on clinical trials, but it's spent in preclinical and basic research. At the same time, drug developments have become more and more costly. So the drug development costs have exploded over the last decades. 2006, it was about $1.3 billion per successful approved single new molecular entity, which is a huge amount of money, and the majority of this money comes from the private sector. At least it came in the time when these figures were generated. Now, how risky is the business? There has been a publication by Ismail Kola, who was actually the person who favors the POC light paradigm. POC was usually an abbreviation for pull out the checkbook, as he called it. So he worked at Merck first, and then he came to Schering-Plough, and in the meantime he works for UBC in Belgium. He looked together with Landis at the success rates for compounds in this process by therapeutic area. So what he found is that from the time when you're first in human to registration, so unfortunately here this can be red because it's black, but this is phase 1 and this is registration. So this big leap here is successful overall in 11% of the compounds. There are areas that are much better in predictability. Cardiovascular, for instance, there we have 20%. Infectious diseases are also pretty good. But areas like oncology, not mentioned here, women's health also, and CNS are below average. And these are figures that come from the time when we had SSRIs, when we had atypical antipsychotics, where we had metabolites or enantiomers of compounds being developed. So these are figures from the golden times where we did monoaminergic drugs, more or less like cookie cutting. What is really worrisome is, and that is something that is really bothering me more and more the longer I work in drug development, is CNS compounds, and they are marked here with the arrows, are pretty good in phase 1. So usually the problem is not so much a clinical pharmacological problem. In phase 2, we are pretty much on average, but when it comes to the translation of a compound that was effective in phase 2, that has shown proof of concept, to translate it into a phase 3 success, which is the bigger, the more generalized population in order to be successful, then we are here about 50%. So it's more or less 50-50. That is clearly something that is worrisome, particularly for executives in the pharmaceutical industry, because here you are placing hundreds of millions of investment and your chance of success is mediocre. So to summarize part number 1, you all know the drug development process of innovative drugs is very complex, resource intensive and expensive, and it has become more and more expensive. Actually, the costs have been accelerated. The public funding of innovative drugs is only a minor factor, so still the majority of drugs that become available for patients worldwide, and particularly in the United States, comes from private investments such as pharmaceutical companies. The R&D budgets in the private sector have increased dramatically in the last decades, but the number of approved drugs has, however, shown a stagnation or even a decline. I realize that this particular slide somehow was skipped in my presentation, but maybe I can quickly go back and show it to you because it's very informative. This one, actually. So while you see here the budgets increasing over the years, you see here in red the number of new molecular entities per year approved by the FDA. And there is a clear mismatch here. You can see on the one hand side this exponential increase of the costs, on the other hand side downward trend in terms of, and this is not neuroscience only, this is all therapeutic areas. It doesn't look better for neurosciences at all, so there it's even much more on the lower end. So this all comes down to what you saw in my first slide, a growing reluctance to make major investments in psychiatric indications in big pharma. And that worries me particularly because, as you heard in a sketch from my bio, that touches upon a core issue that I'm concerned about. Now, what are the specific challenges in psychiatric indications? The first thing I would like to briefly remind everybody is that psychopharmacology is a discipline that developed in the 60s, or let's say in the 50s, more or less by clinical observation and chance findings. Out of a sudden we had a quantum leap. We had antipsychotics, we had anxiolytics, we had antidepressants, which worked in the clinic. They were not rationally developed so much as they were the result of careful clinical observation of a couple of people who followed up on that. But since then, even with the involvement of huge resources in pharmaceutical industry, what we have done basically is to refine the mechanism of action of these compounds, but we actually have not surpassed their efficacy. We have improved the safety in some aspects, but the efficacy has not been increased to a level that everybody would wish for. Still, we have needs for new treatment. I think probably everybody would agree to that when we don't look at the costs, if we look just at the medical need. What is worrisome is that we have made, on the one hand side, tremendous progress in understanding the brain. We have fancy imaging techniques. We can look at all kinds of markers. We have pharmacogenetics, genomics. We have a lot of technologies which were unthinkable at the time when these drugs were developed. Yet, all of this knowledge has not yet translated into novel drugs that are truly different from the ones which we have more or less discovered by serendipity and refined later on. So why is this the case? So what are the main problems? So I think one factor certainly has been for a time that also drug development and drug discovery has very much focused on variations of the theme, monoaminergic drugs, D2 antagonists. But I think that has changed, at least in my experience in industry, tremendously. So I know that people are working on all kinds of targets, even stuff that I didn't even think that people would try to find drugs for. But there are fundamental problems why these things don't advance further, and I would like to elaborate on them. I think one problem, which has been pointed out already for decades, is that psychopharmacological drug development has embraced the nosological or categorical approach, as it is basically the foundation of the DSM classification. I will come back to that a little bit. Also, we have not made as much progress in translational medicine in the sense that it really informs successful drug development so that it holds regulatory scrutiny. Then we have the problem that our trials are so vulnerable because their primary outcome measures are ratings of patients' feelings, emotions, behaviors in different cultural contexts, which strongly depend on the quality of how this is being done by the individual investigator. And also, this all feeds, of course, into the complexity of clinical trials, but there seem to be factors which go beyond that because clinical trials have become more and more unpredictable in their outcomes. I will show you a couple of examples from the Cenopine program, how that can look like, and I was really amazed that this happens really in clinical practice. So I thought a lot about this over the years, and actually I came back to it just a few weeks ago when I rediscovered a book from Herman van Praag, who worked on this already in the 70s, 80s, and 90s, and always criticized the DSM approach as the foundation for drug development, because he said this is an expert opinion-based classification which follows mainly nosological ideas which go back to Kraepelin and other thinkers. Yet the way how we diagnose is we have a choice of lists. We have X out of Y symptoms. You can have very heterogeneous constructs which you finally send under the same umbrella of a diagnose into your trial. And then you go with a very targeted pharmacological intervention which is quite likely to target only a few of them, if at all. So you will dilute by definition in most cases your signal simply by the way how you pick your patients in which you test your drug. And also the same holds true for the biomarkers, because if that is the group you're collecting and you're looking for biomarkers, some patients may have it, others not. It doesn't mean necessarily that the biomarker doesn't work, but it doesn't work for the nosological category, which doesn't really care about neurobiology. So there is this idea, and I'm still struggling with that, and I would be interested in the discussion to hear what you think, that in fact the way how we diagnose is probably one of the major methodological hurdles to make real progress in terms of getting drugs developed and approved over the regulatory hurdles. Why regulatory is such an important thing? That is something I didn't appreciate so much when I was in academia, but now that I have to deal with it, it became clear to me that they have a very important function to actually protect the public from unnecessary drugs. At the same time, their thinking is naturally relying on the consensus of the field to a certain extent, and it is also conservative. So what has happened here is they have broadly embraced the DSM approach and say, well, this is what you guys in the field think is how we should make diagnosis. So if your drugs don't work in the DSM, that's basically your problem. Make a better classification. Well, I think we're far from that. We have to deal with DSM. the spin-off or the collateral problem that comes with it is that whenever you try to define more narrow, cut-out symptom domains, you immediately have to deal with the problem that they say, well, in terms of your diagnosis, this is probably pseudo-specific. Why do you carve out patients with insomnia and vasomotor symptoms, for instance? Who says that that is a valid entity? Why not all insomniacs? So if you have a drug, let's say, that works in vasomotor symptoms and insomnia, but then you have to go for the broader category rather for the specified one where your drug would work best. That is a big problem which has prevented the approval of many drugs in the past. Just to give you an example, I won't elaborate on that, but I was surprised when I joined industry to learn that in regulatory sense, negative symptoms is not yet a valid indication. And we have made efforts with the asenapine program to establish that. Unfortunately, our trials were not that positive that we could pursue it. But there were also methodological problems. We had to prove that this is not a pseudo-specific entity. I mean, I think probably all of the researchers in academia would say, well, there's no problem to recognize that there are negative symptoms, that this is a valid category. Yet in a regulatory sense, it's yet not established. Very similar for cognitive dysfunction in schizophrenia, that there is some way open. Unfortunately, there is no drug yet that has made it through that somewhat opened gate. Now, let me turn a little bit to the problem of biomarkers. So, in the pharmaceutical industry, biomarkers have become a very important tool to rationalize drug development. And it works in some areas very well. At least it gives people the feeling that they can quantify risk. And many decisions in early stages, in order to move them to the costly later stages, are based on availability of convincing biomarker data. Usually up to proof of concept, that is, if you don't have a biomarker that works, probably your program will not move forward. Now, for psychiatry, the problem is that we don't have good biomarkers. We don't even understand how these nosological concepts or even the syndromes really work. So for basically all DSM disorders as defined, we don't have a pathophysiology, we don't have the biomarkers, and not to speak about surrogate markers, where we could use a biomarker in order to predict efficacy of a drug. All that is not available. There have been a number of attempts how this could be made more clear. Or, for instance, this again goes a little bit back to the thinking of Hermann van Praag, not to look so much as the nosological diagnosis. For instance, the green red dot here would be all of the schizophrenias, whatever that means, but to look at components, let's say certain types of working memory dysfunction or executive functioning or mood regulation or aggression or anxiety. And while usually the biomarkers, the clinical models, the animal models don't correlate well with the bigger one, there may be opportunities to validate animal models, biomarkers, and clinical models more closely with subcomponents of the disease. That is exemplified here in the concept of functional psychopathology. For instance, dimensions like cognition, mood, anxiety, volition, drive, hostility, aggression, they could feed into certain syndromes which would be much better targets to validate biomarkers than a nosological diagnosis which consists of a lot of these components which may have nothing to do with your particular biomarker. Besides Hermann van Praag, there have been a number of other groups or researchers trying to specify this. For instance, I recently read a paper by Martin Katz, Charlie Bowden, and Alan Fraser, who basically propose a behavioral component model for depression. And the components which they consider important for the syndrome are listed here. And the dimensions, so more or less the syndromes, the subsyndrome they have analyzed is the anxiety, agitation, somatization factor explaining 28% of the variance, or the depressed mood, motor retardation factor, 26% of variance, hostility, interpersonal personal sensitivity, 21% of the variance. And they also hypothesized that serotonergic drugs act on different dimensions than noradrenergic drugs. That goes a little bit into the direction I'm talking about, to be more specific rather than having this huge, cloudy and heterogeneous diagnostic category as defined in DSM. So what does that mean for drug development? And this is something that also touches a little bit upon my emotions from the last 5 years, because you have to know clinical research is responsible for doing clinical trials, whereas now we have also translational medicine departments in our companies, and usually their focus is actually to get a biomarker in a small study showing an effect. And we had a lot of fights about the value of such a particular experiment. So one of the arguments we always had is, let's say you take such a small subgroup of patients which are, let's call them, enriched, so for a particular feature. Let's say in depression, patients who have particular anxious or hostile elements, because you want to study an effect of an SSRI on that. That has quite a likelihood that if your intervention is right and your biomarker is right, that you might see a signal. What happens then often, unfortunately, is you say you have proof of concept, and then you throw it into phase 3, where you go back to your nosological category, where you cannot focus on the enriched population because that's not a valid developmental diagnosis. And then you may have your failures, as you have seen in the statistics, which explain why a very sensitive experiment in phase 2 doesn't translate in success in phase 3. So an important question, in my view, is how can we make sure that dedicated, focused, and necessary trials in phase 2, which obtain proof of concept, actually translate into development success in broader categories as they are requested now. So, I've spoken a little bit about the concepts. I've spoken a little bit about translational medicine and the problems which actually happen in real-life drug development. But I mentioned that there are other factors which are much more subtle or soft and actually may have an even more detrimental effect on the success of drug development. And these may be factors that have particularly to do with the way how we do the trials and how we have to rate the symptoms, the selection of patients who are in the trials, the people who actually do the trials, and also the whole business of drug development, with its dynamics, complexities, which sometimes are not taken into account as a factor influencing the outcome, because, remember, the outcomes are subjective measurements. And finally, all these factors may have overall a contribution to the risk-benefit assessment. That is a term more and more used, not only by European regulatory agencies, where that has been the mainstay all the time, but also the FDA more and more comes to risk-benefit assessment. Okay, you have 2 trials, so you have 3 positive trials, but what is the overall assessment? And the safety assessments become more and more critical. The FDA is more and more sensitive about any kind of safety signal, and therefore the risk-benefit assessment may change even in instances which 10 years ago may have not been a problem for approval. I have to touch briefly, of course, on the placebo effect and what we have seen here over the last year. So it was not a surprise to me that in depression we have a placebo response. The worrying thing is that data indicate that it was ever growing, but the same now happens also in schizophrenia. And I myself and my company, to which I have been loyal, and depending on which one is the real company I've been loyal to, but have been subject to this problem, an increasing placebo response, even in trials with acute schizophrenic patients, where I would have thought from my clinical experience that these are not really the patients who react very well to a non-specific intervention. And problem is we don't really understand what makes placebo such a problem in the trials, even in these formally safe indications for drug development. Now, you probably know this slide from Walsh from the JAMA publication, where you can see that over the time the response to placebo increased on average. Unfortunately, we have seen the same also in schizophrenia trials, so here you have to read it in a different way. This is actually the PANSS change from baseline, and you can see it over the years it became bigger and bigger and bigger. And I can show you here examples from the asenapine program. This was a phase 2 trial done at the beginning of 2001 in 20 American centers. You can see approximately 5 points of placebo response with on the primary efficacy parameter, and a very nice and robust response with asenapine, which clearly and with no problem separates from placebo, no matter which method of analysis you're looking at. Now, 5 years later, 50 centers, bigger trial, double the size of patients, twice the placebo response, about 10 points. And you see the drug does more or less the same as before, but here it becomes more and more difficult to separate. In fact, here the 5 milligram separated significantly from placebo, whereas here the 10 milligram, just a little bit above it, did not make it, which meant in the label it didn't end up as a dose effective for the patients. Just very few. Would we have chosen the MMRM, we would have made it. Basically because we have this very pronounced placebo response here. And even worse, we had trials that completely failed. So you see here a flexible dose trial, asenapine flex dose, olanzapine flex dose, placebo. Almost identical results. This is very scary. So this was the first trial actually that was finished while I was in charge at Organon. So I really thought maybe it's, I thought, well, hopefully it was not my fault. It turned out not to be, but of course you get scared when you see this. I mean, this is totally clinically counterintuitive. I'll come back to try to explain a little bit what maybe the reasons for this. So certainly I think there is something like a true placebo response, and there is also neurobiological research showing that there may be a number of mechanisms which mediate that. But the important thing is, I think that placebo response is not necessarily a response to placebo. I think this is very important to realize, that whatever you measure there, it's not necessarily a true phenomenon. It may be an artifact. And the artifact part of it, that is something we need to get a grip on. We need to get a grip on measurement errors and artifacts, for instance, like baseline inflations, expectancy biases, rater drift, inadequate performance, but also inclusion of the wrong people who then, of course, behave in a very non-specific way. That is something that I became more and more concerned since I work in industry, and I learned how important these things are, these so-called soft factors. So this slide summarizes what I think the 3 major areas where we need to work in order to secure a future for drug development in psychiatry. The first one is a very pragmatic one. We need to do an effective quality control of the diagnostic and also the severity assessment during the trial. It is actually surprising, looking backward and having had the bad experiences, that we actually really thought that we could handle a trial by doing a rater training at the beginning of the trial with all those 100 raters sitting in the room watching a video and saying, well, how much depression do you think this person has? Well, it's 3. No, it's 4. Oh, it's 4. It's 3. It's 4. Okay, finally you agree. You pass your exam. And thereafter, nobody looks anymore. What do you do when you rate these patients in real life? Do you really rate them? Question number one. Who knows? I mean, there is something in the CRF written. Nobody has seen whether someone has really done an interview. Number two, how much skill has someone put into elucidating information that is necessary? We have no surveillance on the performance and the expertise and the quality over time. Then raters change. Rater number A does the first interview. Rater number B does the second one because rater A is on vacation or something. Out of a sudden you have a drop. Then the next rater comes back and rates again according to his standards. You go back. We have seen all these kind of things. This is something that is totally unacceptable for the future. We have to fix this problem. This is not a minor sideline of drug development. Point number two, translational efforts need to span the entire drug development process, while the current focus is mainly between preclinical and proof of concept. And sometimes I joked with my colleagues, you folks from translational medicine, once you have proof of concept, you throw it over to us and you think, now it's on autopilot and it will win. And if we don't win, then the clinical research guy is the stupid one who messed it up. That is just not the way how it works. The third one is probably, I spoke with John this morning, a lifetime effort, which I hope I will witness it, but I'm not sure. We need to, in a way, come to a more rational way how we approach the target symptoms which we want to treat with drugs and for which we want to develop drugs. I think this is a very difficult process, but finally, I think there's a lot of value in biomarkers, and if we want to follow that route, we have to validate them in a regulatory sense, not in the sense that I've done a study with 30 patients and it worked, the marker is validated. That is not validation. It has to be something that prospectively predicts the outcome of the treatment, more or less in the sense like cholesterol finally made it to a surrogate marker because it showed, well, cholesterol plays a role in cardiovascular mortality. Lowering cholesterol is a target for the drug, so therefore you could show that it makes a difference for the clinically relevant outcome. I think a very nice example is the ABNEY initiative, where I learned a lot when I was in charge the last 2 years for the Alzheimer program, how much groundwork has been done here in the last few years. So a prospective assessment of a couple of hundred people looking at relevant biomarkers provided a huge amount of information which informs the people in industry with their different amyloid-based approaches to do relevant clinical trials to finally test the hypothesis. Is it A-beta 42? Is it phospho-tau? Is it total tau? And how do they change? Is hippocampal volume really something that can monitor the progression of the disease or not? There are a number of testable hypotheses derived from empirical research which you can really address in an adequate and dedicated trial with a nicely designed drug. I think we need things like that in psychiatry as well. So just for a couple more comments on some of the factors. I mentioned the fact that there are factors related to patients who actually enter our trials. I mean, this idea of professional patients is really not a myth. We have seen many instances in our databases, actually patients who have been in the same trial in 3 different centers in a row, and the connecting dot was a bus line in California from one center to the next, and things like that. There are many examples which are really scary once you look into this. This is from our asenapine database and shows you a very interesting result. We have done the phase 3 development in acute schizophrenia. The protocol said patients had to be acutely exacerbated and have a diagnosis of schizophrenia. Now, we thought that's good enough. It turned out that approximately 1/3 of the patients had an acute episode of more than 6 months duration once we analyzed the database. So then we analyzed what happens to the drug placebo separation if you take only the acute ones and you take those with the 6-month duration of the episode. Now here you can see what happens to those who actually, how should I read this? Here are the bottom line is the acute patients who had a sufficient severity of at least 80 points on the PANSS total at entry were the ones that truly made the drugs work in the trials because they separated from placebo nicely. And that happened for asenapine and also for the comparators. This was not drug specific, but it happened with all the drugs that worked. Once you had patients that were not severe enough, less than 80 points on the PANSS, or were not acute enough, so chronic patients, the two groups basically superimposed and you had no separation between active and placebo treatment. Bottom line is, if you get the wrong patients in your trial, there is probably no way how you can really save the trial. When looking at the investigators, this has become more and more of a concern. I think it is one of the most crucial things to pick people who do a very good job, who have the right motivation to do it, who are quality committed, and who are also qualified enough, expert enough to do a good job in making diagnostic assessments and rate the patients over time. And also you really need to control for a consistency of ratings across centers. Not everybody has to be the most perfect and sophisticated rater. The most important thing is that people rate consistently over all your centers all over the world. And that is already very challenging, and it needs to be monitored. It needs to be controlled while the trial is going on. In fact, we were so worried about it at Organon that my little team at that time started together with the cognition group to set up a pilot project, which actually worked out pretty well. And in the meantime, a lot of CROs have adopted this approach. What is actually happening there is the site investigator still rates the patient as it usually happens in the trial. But at the same time, this whole process is monitored by a webcam and recorded. And this record is being sent to a native-speaking expert who independently, within 48 hours, has an independent review of the video and send his scores back to the site. So the site can either ignore him or adapt according to him. In any case, we had 3 databases: site, expert rater, and adjusted site rater. And we found that it improved in about 20% of the patients substantially the concordance between the two, and the trials also ended up nicely. But one important thing is there is a supervision that actually a real interview happens, and you can also monitor the rating performance. You can see how actually the information is being elicited, not just that someone has made entries in a case report form. And I think many CROs now focus on this aspect and try to calibrate it to make it better, to make it concordant, or to have centralized raters, such as the Medavante company. So there are a number of very important initiatives which try to fix this problem. Well, baseline inflation, I think I spoke about it. This particular slide I wanted to show you to make the point about the translational efforts and where they are centered. This is the program of a large Dutch and European consortium called TI Pharma, which looks at validating biomarkers, particularly for depression, but also for other indications. And the point I want to make here is, you see here the whole sequence of drug development. All these arrows here center up to phase 2. And then it says, financial power to finance clinical trials beyond the scope of TI Pharma. Basically stays with the companies. But that is actually where you lose the battle of approving and getting the drugs approved. So you can be very sophisticated here, and this one shows it even a little bit better. You see a lot of translation forward and backward up to proof of concept. But you have to make it up to here. And there is not a lot of activities happening here, because this is usually not very fancy. You have to deal with site raters and complexity and operations and so forth. It is scientifically much more interesting and challenging to work here, but if we are really serious about developing a drug that gets to the market, we have to make the full mile here. Okay, and finally, this is my last slide. I want to once again talk about ABNEY and say that I think this should be kind of a model for further initiatives, that people from various stakeholders, including NIH, NIMH, academic centers, and the whole bunch of industry interested in Alzheimer research, get together, find a way how they combine funding efforts and do a prospective study that generates the data that are needed in order to do the more rational biomarker-based work that actually everyone looks for, particularly in the Alzheimer area. I think in psychiatry probably we need a little bit more work on what exactly are we targeting, but I think this kind of effort will be inevitable if we want to do a more rationalized drug development. Thank you very much.
Questions?
My name is Inayat Khan. I'm from the Safety Surveillance Group. You alluded to a very dramatic decrease in the investment in the RDU by pharmas in the psychiatry area. So what's the reason? I mean, are they losing money in their sales in the psychiatric medication? Or are they obliged anyway to allocate certain percentage of their income? So why they have cut down so much?
By nature, I don't know exactly the reasons for that. I can only speculate. I think you have to see in the big pharmaceutical companies, there are several areas competing for internal resources. Pharmaceutical companies are business corporations who have to generate profit because they are usually owned by shareholders who every quarter want to see good figures. So you are under tremendous pressure to do innovative compounds that have a chance to make a difference on the market. At the same time, you have to have a high probability of success to get those allocations of resources. And as I showed you, you're competing with areas which have a higher probability of success. So in some companies, the decision may be what's coming up the pipeline in neurosciences. And they say, okay, we have the amyloid hypothesis in Alzheimer's. That looks pretty good. It's testable. So there are a lot of compounds out there, so maybe that works. Okay, let's go there. What about depression? Well, 60-70% of the trials fail anyway. Why? We don't know. Okay, next compounds. Well, we have a couple of serotonin antagonists or so forth. We have some high-risk projects. Probability of success, 30%. Next one. You understand what I'm saying? So it may be the case that they just don't have the confidence that the way how we are developing drugs will be successful on the short term. My personal speculation. I don't know whether this is true, but the fact that this happened, as I showed you in the first slide, within 6 weeks in a couple of major companies, indicates that this is probably a bigger worry and assessment of a risk and a risk aversion which has developed, which is different than a couple of years ago.
So thank you very much for that. That was a very interesting and challenging, thought-provoking, and not so happy-making talk. So in every glass, there's which looks half empty. There's actually a lot of opportunity, and one of the things that seems to be happening is an acknowledgement that the blockbuster model, which is essentially the low-hanging fruit for the group average effect, is going to have to shift toward stratified medicine, so that we're running trials in patients who are actually likely to benefit. the problem with that is to get to stratified medicine, we need to develop the tools for stratification.
Yes.
And a large chunk of that bet is going to be placed on biomarker or biosignature development. So that in turn is going to take collaboration. So can you talk a little bit about how you see Merck or other companies moving toward this stratified model and doing it in a way that involves helping to build these consortia to solve problems which are held in common by all the companies?
Well, first of all, thank you very much for a fantastic summary of my lecture with a positive spin. I should have kept it briefer. So what I can say to my joy is that at this time, Merck has publicly committed to neurosciences, which includes psychiatry indications, some of them, particularly the focus of schizophrenia, and also circadian disorders, which has partially to do with insomnia, where they're working on, but also depression is still something that is being contemplated because it's perceived as maybe linked to circadian disorders. We have a so-called neuroscience franchise, which has a critical mass within the company right now. And Alzheimer's disease is something we're also interested in. So I think that the way how I perceive Merck is once they commit to something, they do it earnestly and with all efforts to make it a success. And a very important component in this is our sound bilateral cooperations with whoever delivers value in a corporation in order to make the process a success. And I think collaborations with academic institutes like DCRI, for instance, is certainly a possibility. But it goes beyond that. The ABNEY process is something that has been supported, and if things would come up in other areas, I'm pretty sure that Merck would be part of it. It depends, of course, on the project, but in principle, there is a very scientific mindedness in the neuroscience franchise, which I enjoy a lot, actually.
So, Armin, let me ask you another question. We've talked a lot about whether we ought to sponsor or host a workshop that kind of aims at method variance in clinical trials in psychiatry, the kinds of things that you talked about with baseline inflation and rate reliability, issues within experimental design that decrease signal detection. There hasn't been one of those. We actually used the radar calibration method that you talked about using video in the TADS trial and had a 34% placebo response rate for the fluoxetine placebo drug difference. So those kinds of things people don't like because they're expensive and they're cumbersome. They take additional effort and structure. But I think they're absolutely essential. So would you think that Merck might be interested in participating in a methods workshop that involved the FDA, other companies, academic experts, where we'd kind of put together a standards kind of discussion?
I'm pretty sure, yes. I mean, as far as I'm concerned, I'm more than interested. And I know that David Michelson, who is the head of clinical, also acknowledges there's a problem. I mean, we are very committed. If we are doing big programs, that we have to control this factor. If it costs additional money, in the meantime, I think the thinking has shifted. That is well-spent money if you do it right, because you're protecting a major investment and you're making sure that you're testing the experiment in a way that delivers meaningful answers. That is the most important thing. I think yes, if we would have that opportunity, at least I will be there.
You also brought up a very good point about picking the right sites and the right subjects. So, having said that, what is your theory of how to pick a right site, right subjects? I mean, we talked about biomarkers, validity, but there's more to that. So,
I mean, this is really you caught me on a problem that I haven't solved yet. My views have changed over time. I mean, depending on what kind of bad news came in, I changed the course, but I thought I have to have a more comprehensive theory of it. It certainly has to do, first of all, a site is only a physical location. I think it's most important who actually really performs the work there. If I do a rater training with a principal investigator who is brilliant, but actually never sees a patient, but in fact he has very junior people who have no clinical experience in doing the ratings, though they have passed this test of 30 minutes looking at the video, we have to disentangle that, first of all. Also, I thought for a while that past performance, good performance, is a predictor for good performance in the future. Now, having talked to a number of people, that is not so predictive. What is predictive is if someone did a crap job in the past, he is most likely to do a crap job in the future. So don't pick those sides.
I would want to speak a little bit about site selection in the sense that, one of the influences that sites get from pharma, of course, is to enroll so quickly and that the timelines are so critical. And I think ultimately we're all going to have to step away from the time is money mentality a little bit and focus more on good patient selection. And I wonder if you're seeing that kind of thing done in pharma. And at the same time, I used to actually run a couple independent sites, and I saw a lot of pressure myself to go from maybe 50% clinical, 50% research to 10% clinical, 90% research, and I saw the tendency to have more professional patients coming in the door. When we look at site selection, a lot of it, maybe even more important than who are the investigators and how senior are they still affiliated with a strong clinical operation, or have they become so focused on the research that they're now getting patients that they're not familiar with, patients that don't have much affiliation with the site, and just bringing in people predominantly through advertising. And I wonder if you have experience with that as well.
So to your first point, I think there is a growing awareness in pharmaceutical companies that the problem has also to do with the type of patients, the type of model, the time pressure. So I hear more and more people advocating internally that if we go in a certain indication with a project, we have to allow a reasonable amount of time to get quality recruitment rather than just to get warm bodies into the Excel sheet and then ticking off the box, study done. There are more and more people listening to that. In each and every instance, it's important to, again, educate all the people involved in decision-making, because you have to realize sometimes decisions are made by people who are not CNS experts, who have different experiences in other areas where it doesn't matter so much, where, in fact, patient selection is not the decisive factor. I have been a prophet all the time, and with me a couple of other people, and I hear more and more people making that point. I'm very happy that David Michelson now is also a very strong advocate for quality. So there are people, and there is more and more recognition for it. But there is also a time pressure, because project management is in charge of driving the project to a successful and also as quick as possible end, because time is money to a certain extent. So you cannot prolong that indefinitely. So we have to find a way to strike the balance to get quality recruitment and still a fast processing of the study done. This is very difficult. I don't know how exactly that looks like. Remind me of your second point.
Well, just the issue with sites still having some clinical affiliation. That would seem like the number one site selection criterion in my mind.
Well, yes and no. And depending on what database you look at, sometimes this is a factor, sometimes not. Even in academic centers, sometimes people are so busy with getting all kind of studies to the house, and they have little time to actually take care of their conduction and finishing. So sometimes we find that high-profile academic centers actually do very poorly in terms of the actual trial performance. And sometimes sites that do it as a business model are very good because there is someone sitting who cares and has the right attitude. So I think, again, it's unfortunately a bit more gray than black and white.
Anybody else? Okay. We very much enjoyed having you. Thank you for coming.
Thank you very much. Thank you.